Within nature, parasites have found countless ways to survive and thrive off of different organisms. One of the most notable characteristics is the ability of some parasites to change the behavior of its temporary host, or intermediate host, so that it is more susceptible to predators. This behavioral change enables the parasite to enter into the host that allows it to undergo sexual reproduction. This organism is known as the final host. A famous example of how host manipulation is used for the purposes of transfer is carried out by Toxoplasma gondii. Toxoplasma gondii is an obligate intracellular parasite and is part of the Apicomplexa phylum and Sarcocystidae family. This parasite is very common even in the developed world, and felids including domestic cats are the only known host where sexual reproduction can occur. How does this parasite find its way into cats? It is ingested as an oocyte by rats, where it then manipulates the rat's behavior, thereby increasing the probability of it being killed and eaten by a cat. A 2015 study by researchers in Singapore and from Duke University presents data on how Toxoplasma gondii may influence such changes. Researchers used adult male rats, one control group injected with saline, and one group injected with tachyzoids, a cellular stage of Toxoplasma. The rats were given a choice of two levers, one with an immediate payout of one food pellet, while the other provided 4 pellets after a variable delay. Researchers found that the rats infected by Toxoplasma were less tolerant of the delay, suggesting an increase in choice impulsivity caused by the parasite. In addition, researchers examined spinal segments of control and infected rats, finding a decreased number of neurons from the nucleus accumbens in the infected group. The nucleus accumbens is part of the basal forebrain and plays a large role in mediating choice impulsivity. Further analysis revealed a decreased amount of dopamine present in the nucleus accumbens core of the infected rats. All in all, researchers propose that these findings are consistent with the behavioral syndrome that reduces innate fear, increased sexual attractiveness, and delay aversion, leading to a carpe diem mindset in the infected rats. Questions still do remain over how Toxoplasma alters dopamine levels in certain brain regions, including data that suggests an increase in dopamine rather than a decrease in infected rats. Nonetheless, the research presented shows evidence of host behavioral alterations by Toxoplasma gondii, which are crucial to its survival.